Now we are going to study southern blotting. Southern blotting is discovered by E.M. Southern scientist. The principle involved is southern blotting is used for the detection of DNA, and the principle involved is firstly separation of DNA by gel electrophoresis, and then detection of DNA by label probe hybridization. It is similar to ELISA but a slightly different. The principle is gel electrophoresis method, by which DNA is separated and then detected by using label probe hybridization. Now procedure. Firstly, we should take a DNA and then it is cut by using restriction endonuclease. The DNA is broken and then the broken DNA is treated with NaOH for its denaturation. Fragments of DNA are formed. To the fragments of DNA, agarose gel containing buffer is added. Now this preparation is sample. This sample is used and then it is immobilized. For the immobilization, we use nitrocellulose for the blotting. Blotting is nothing but immobilization of desired DNA or protein. So for the blotting, we use nitrocellulose membrane. The blotting technique is flowering. Firstly, filter paper is placed and then this sample is placed above it. Nitrocellulose is placed and then 2 to 3 layers of filter paper is placed. After this preparation, sample reaches the nitrocellulose membrane and goes up to filter papers, but the fragments get trapped in the nitrocellulose membrane, and then this nitrocellulose membrane is exposed to a solution containing labeled cDNA probe. Then autoradiography is performed. This is the procedure. So first we need to take the DNA. It is cut using restriction endonucleases and then NaOH is used. First it is only broken down, but then NaOH is used to make it even more fragmented forms. To this fragment, agarose gel with buffer is added. This preparation is sample and then it is immobilized. The immobilization done with the help of nitrocellulose, and the method involved is flowering. First, on a filter paper, sample is placed and above it nitrocellulose solid matrix is placed, and above it again 2-3 filter papers are placed. In the sample solution moves upwards, but the fragments get trapped in the nitrocellulose membrane. Then this nitrocellulose membrane is exposed overnight to a cDNA probe, and then autoradiography is performed for the detection. This is the procedure involved in the southern blotting. We can revise it again. The principle involved is gel electrophoresis and then detection of DNA by label probe hybridization. The procedure involved is nothing but DNA is cut, restriction endonuclease by using restriction endonuclease, broken down DNA, treated with NaOH, fragments formed, agarose gel added. Now it is sample. Blotting is done using flowering using nitrocellulose matrix. The sample moves up but fragments get stuck and they are exposed to cDNA probe, and autoradiography is performed. Now applications of southern blotting. Southern blotting is used for gene analysis. It is an important method for the confirmation of DNA cloning. Restriction sites mapping can also be done, also helpful for zoo blotting. So these are the 4 applications: gene analysis, DNA cloning, restriction site mapping, and zoo blotting. Used in gene analysis, important method for confirmation of DNA cloning. We can do Restriction site mapping also helpful for zoo blotting. So the applications of southern blotting involves gene analysis, DNA cloning, restriction sites mapping, zoo blotting. Important method in DNA cloning used in gene analysis, also used in restriction sites mapping and helpful for zoo blotting. Thank you.